So again, welcome everybody to this webinar about our spring grant program. I'm excited today to talk with you about, about these new grants. And I'll remind you that the mission of FPWR is to eliminate the challenges of PWS through the advancement of research and therapeutic development. So we support a broad portfolio of high-impact research, and we do that across the translational spectrum. So everything from discovery, understanding the basic science of PWS, through clinical trials and optimizing clinical care. And we do this in two major ways. So we have a directed, what we call a directed research program and research tools. And this is where our research team, which is shown here on the right, working with experts in the field, identifies areas that FPWR can invest in that will help the entire field move forward. So sometimes these are animal models, sometimes this is infrastructure for clinical trials, these kinds of investments that help the entire field. And these programs are managed by FPWR's research team. The grant program, which is what we're going to focus on today, is what we call investigator initiated. So basically, we set aside a portion of the budget and we ask scientists and clinicians from around the world to send their best ideas about what needs to be examined in PWS and you know, how we can improve therapies and the quality of life for individuals and their families. And we evaluate those ideas through a competitive grant program. And we do that twice a year. And so we'll be talking about the grants that came in last fall and have just recently been approved for funding. So again, when we think about the research spectrum, we go all the way from discovery through drug development, through clinical trials and into post-approval, or where we try to optimize the treatment for the person with PWS and approved treatment. And as I mentioned, our directed programs are shown here at the bottom, and they are at selected points across this pathway to try to accelerate the research across the entire pathway. In contrast, our grants program is really anywhere along this pathway. So in our grants program, we fund anything from basic discovery all the way through early phase clinical trials. We don't have enough funds to do large stage clinical trials, as well as optimizing care in the post-approval phase. So the difference for our grant program is we're looking for innovative research, exceptional opportunities, usually somewhat higher risk early stage projects. And that's what we'll be discussing in this webinar. But before I do that, I just want to kind of interrupt and mention to everybody, in case you're not aware, that 2018 is going to be a really, I think, an amazing year for our community. So for years, I and many others have been saying we really need some new therapies to help our kids manage their hunger and their emotional volatility, so their hunger and their behavior and all of these things that impact on PWS. And well, you know, the time is now. So we are really fortunate that there are going to be several clinical trials testing new medications for our children and adults with PWS. And it's truly an exceptional time in the history of our field, I think. And I'm very hopeful that we'll find some new things to really make a significant impact on the quality of life of PWS. But it is going to take a lot of work on a lot of people's part and a lot of participation by our community. So I would encourage you to start learning about these clinical trials. There's a website on our website. You can find information on PWSA's website. You can find information about the new medications that are going to be tested, and start looking at those and seeing if any of those might be a good fit for you and your loved one with PWS. If you are not eligible for those trials, or if you don't feel like those trials are right for you, there are many other ways that you can become involved and push the entire clinical research forward. On our website, we also have non-medication trials. So for example, if you have an adolescent who is having anger management issues, there is a mindfulness intervention to see if we can teach individuals with PWS mindfulness. And that's a remote intervention. So the study is funded in Australia by FPWR, but you can participate from anywhere in the world. So please become familiar with these opportunities and see where you might be able to contribute. The Global PWS Registry is something I hope everybody has registered for and provided information so that we can understand the natural history of PWS. We're going to be using the registry for additional studies in the coming year that I think will be very exciting. We have a weight project that will be debuting next month, where you can send us your child's weight for those 12 and above for a period every week for a period of 6 months. So we can just look at what is the natural variation in that weight in people who are not receiving medication. So we have something to compare to as these new medications come online. So finally, we have an opportunity next month in which we're going to partner with PWSA and give a joint webinar to answer your questions about clinical trials, the clinical trials in general, give you an overview of the clinical trials that are going to occur in PWS, and hopefully give you a lot of information about how to get your questions answered so that you can make the decisions about potentially participating in some of these opportunities. So please sign up for the clinical trial alert, look for the webinar announcement and register for that, and we hope to see you at that webinar as well. So, okay, diversion completed. We'll go back to the grant program and the work that we're going to fund in the first part of 2018. So as a reminder, the goals of our investigator-initiated grant program are to support early-stage, high-risk innovative research. We select grants on the basis of both scientific and parent advocate reviewers. So in that way, the scientists review the scientific merit, the parents evaluate the importance of the project for the PWS community, and working together, we select those projects that are scientifically sound and are likely to have the biggest impact in PWS. And we really focus on research that aligns with the priorities of advancing our understanding of PWS and developing effective therapies. Another really important aspect of this program is to expand the base of PWS researchers, so those who might have expertise in other areas that are important for PWS, such as mental health or obesity, and bring their expertise into the PWS field. We also use this program to support promising young investigators and to support investigators who are well established but who want to have an idea about something new to try. So our grant program has been around since FPWR was established. We gave our first grants in 2004, and since that time we've invested more than $10 million into the grant program, supporting more than 150 projects in 11 countries. I think it's important that, especially in a rare disease, it's important to go where the best research is. So we support research internationally. We're just looking for the best studies across the globe. One of the most important ways that we measure whether the investments that we're making are having an impact in the field is based on the publications that come out of those studies. So scientists publish, and that's their way of communicating to the rest of the scientific community the findings that they have. It allows others to build upon their findings and snowball the research. And we, again, reflective of the kinds of things that we have supported in our grant program, we've had publications all the way from gaining new insights into the function of genes in the PWS region, to understanding appetite, developing new drugs, testing drugs and interventions, and improving clinical care. Importantly, one of the goals of this grant program is to get projects started that can then be competitive for additional funding from, for example, the National Institutes of Health in the US. And our investment of $10 million has resulted in more than $10 million of additional funding as these projects have grown and the investigators have gotten additional funding from outside of FPWR. And it also is important for developing new resources such as cells and animal models that can be used across all PWS researchers, new drug targets, and new insights into clinical care. This gives you some statistics on our grants program over the years. You can see when we started back in 2004, you know, our funding was relatively low, reflecting our modest starting point, but has really grown over the years, especially with the One Small Step platform, and we really have gotten to a point now where we're funding anywhere between $1.5 and $2 million per year in our grant program. You can see we had a little drop-off in 2016 and 17. That's actually not a drop-off. That's when we started doing our directed research program. So some of the projects that would have been in our grant program were shuttled into that program. So you can see on the right the diversity of the kinds of projects that we support through this program, which really reflects the diversity of issues in PWS. And you can also see the proportion of research that's done in kind of straight up lab dishes, which is in the blue and in vitro, in animal models, which is when we move things to usually in preparation for testing them in humans. And then a growing portion of our research is now the clinical side, so research that involves people or tissues from individuals with PWS, and this has really changed over the years. It used to be about 20% of our research budget. Now it's almost 40%, and I think that reflects the maturation of the field and the maturation of our organization. As I mentioned, we try to bring new investigators into the PWS world, and about half of those who we fund did not have PWS research funding before. One statistic that I think we really need to improve is that over the last 5 or 10 years, funding overall has become much tighter, and so we have a larger proportion of senior investigators seeking our small pilot funding, which is great on the one hand, but I think we need to be careful that we're not we're not fully supporting our junior investigators. So I think we need to improve on that statistic. So I just want to give you a few examples of our returns on investment so far. As I mentioned, publications are really important, and this just shows some of the publications that have been fully or partially supported by FPWR since the beginning of the year, and you can see there are many of them. I'll just highlight a couple. Dr. Holland, who has received FPWR support and is developing a vagus nerve stimulator to improve behavior in PWS, also is an expert on all mental health aspects in PWS, and he's just published a very nice paper laying out some of the neural pathways that might be impacting this very high risk we see in PWS of developing psychosis. This is an important question, obviously, and this paper lays out some hypotheses. It synthesizes the information that has been gained over the last few years. You may remember that we held a mental health workshop in PWS, I guess about 5 years ago now, which we'll be updating on our website to let you know the progress, because there has been a lot of progress on the top priorities of that workshop. But one of the top priorities is really understanding this high risk of mental illness, and Dr. Holland really lays out a very nice arguments about the pathways that are likely to be contributing to that. A second study I want to mention is one from Dr. Yao's lab at Cambridge. One of the issues in PWS research is that one of the most widely used mouse models, which has a deletion of the SNORD DNA, the SNORD genes, the mouse model doesn't really mimic the human condition, because it mimics the fact that they're born kind of small and have failure to thrive, but that mouse model never becomes obese. And if you don't have a mouse model that becomes obese, it becomes very hard to test anti-obesity drugs in a PWS model. So Dr. Yao's lab used a genetic approach to knock out expression of this gene after the mice were born. And this allowed a portion of those mice to become obese. So for the first time, we have a mouse model targeting SNORD116 that develops into an obese mouse. And we'll hear more about that in our new grants, because we're going to continue to support that work with Dr. Yao. A final example of some of the returns on investment that we've seen already this year is the work out of Dr. Lalande's lab, which is now being carried out by Dr. Chamberlain, since Dr. Lalande is now the Vice President of Research at Shriners Hospitals. But this is work that has been going on for a number of years, looking at ways to activate the usually silent genes on the maternal chromosome. So we first supported Dr. Lalande to look at the machinery. So how does that maternal chromosome get silenced? And in deciphering that, he identified this particular protein, zinc finger 274, that regulates this process. What he showed in this new paper is that if you get rid of that protein, you get re-expression or turning on the genes on the maternal chromosome. And this is important because all individuals with PWS have that maternal chromosome sitting there, whether they have deletion or UPD. And so this is potentially a therapeutic approach to restore normal gene expression. And this complements some work that we supported along the way from Dr. Zhang's lab, who's using a drug approach. So this was a genetic approach, so a gene knockdown approach. This is a drug approach to achieve the same thing. So slightly different approaches to achieve the same ends of activating gene expression. I think at this early stage in the game, it's really important to kind of leave our options open and explore different ways of getting at this problem. One thing that this and other studies that we're supporting on gene activation for PWS allows us to do now is, us meaning them, allows them to do now is start looking at feasibility questions. For example, would an adult with PWS, if you turn the genes on when they were adults, would that have an impact on their symptoms? Or does it have to be done, for example, when they're a baby or in utero? And we don't know those things yet, but now we have the tools that we can ask those questions. And so I think gene activation is an exciting time in PWS research. We're a long way from having something in the clinic, but we see a path forward. These studies have shown that you can activate that maternal gene, and then now we have to address these feasibility questions. And FPWR will be emphasizing that in 2018. We're planning a request for applications for the latter half of the year to look at some of those issues. Okay, so now the new grants that we've approved for funding this year, the list of them is shown here, and I'll just give a little brief overview of them. So we have two projects that are looking at the genetics of PWS. Both of these are really focused on that critical gene or set of genes, the SNORD116 gene cluster. We have two projects that are looking at the cellular biology. So what are the disruptions in cell biology in PWS? We have a animal model project, one on the gut microbiome, which is really interesting. And then finally, a project improving social function. So we go all the way from genetics all the way through to PWS social functioning, again, which is how this grant program is supposed to be. So to look at this a little bit more closely, the first two projects are really getting at a very similar question. What is the normal function of this SNORD116 gene cluster? We know this is a critical set of genes in PWS. If you lose this small set of genes, you get most of the characteristics of PWS. So they're clearly doing something important. And the goal here is to identify what those cellular processes are and identify new drug targets based on that information. Now, many of you will know that we have made a lot of progress in this area in the last couple of years. Other groups have been looking at this. Janine LaSalle and her group has shown that these snoRNAs are important in regulating sort of the circadian rhythm of gene expression, and she showed that a couple of years ago. It continues to pursue that angle. Dr. Leibel and his colleagues have shown that the SNORD116 genes may influence the expression of PC1, thereby impacting hormone processing and causing some of the endocrine problems in PWS. And Dr. Stamm, as we'll talk about in a minute, has shown a function in RNA stability. And all of those studies are continuing to progress, and these investigators as well as others in the field are developing support or not for those various hypotheses. But I think as a foundation, it's important for us to continue to support many different studies, kind of many ways to look at this same issue, because many of them will be complementary and will reinforce each other as we really understand the full function of these snoRD116 genes. So Dr. Stamm has been looking, he looks at there are SNORD genes in different places across the genome, and he's been looking at how they impact RNA splicing and stability. And he's found that they play a role in these really important functions, and particularly when RNAs are expressed kind of in a spiking pattern. So this does relate back to Dr. Leibel's project and Dr. LaSalle's project, in that those RNAs that are on a circadian rhythm and RNAs that are processing hormones, are oftentimes kind of tightly expressed. And his data shows that these snoRNAs might be controlling that tight expression of those genes. And so the goals, and he's been looking at this over the past year, and the goals in the upcoming year are for him to continue his work determining how these snoRNAs influence RNA stability, how exactly they interact with their target RNA. So they are not interacting with just any old RNA, they're interacting with certain targets, and determining where they act in the cell. So most snoRNAs work in the nucleus of the cell, but Dr. Stamm believes that these snoRNAs may be acting in the cytoplasm, and that's important for understanding how they function. So we're excited about this grant because Dr. Stamm is a productive investigator who knows a lot about RNAs, and he's defined this novel function for SNORD RNAs. He's using advanced technologies to answer these basic questions, and the potential long-term contributions are to really understand how the loss of snoRD116 leads to the PWS characteristics, identify some of the downstream targets, and also to develop strategy to replace the missing function in PWS when we lose the snoRD116 genes. Dr. Bratkovic is a young investigator who's using a somewhat different approach. So he is using some new technology that looks at how RNA interacts with RNA across the entire across all the RNAs in a cell, which is called a transcriptome. So he's looking at the interactome, everything's an ome these days, the overall interaction of those snoRD116 with any and all RNAs. And there are some new technologies that are being applied to those questions, and he's developing them specifically for snoRD116. So this again is a second year of funding. In the first year, he did some preliminary work, evaluating these different systems. He's refining the strategies and the technology to identify the full complement of snoRD116 interacting RNAs. And then he's going to validate the functional significance of those interactions. So you want to know that not only is it interacting with the RNA, but it's actually having a consequence, and what is that consequence? So we're excited about this grant because it's a young investigator. As I said, we really need to, I think, boost our support of young investigators. And it's a young investigator using brand new technology to address the question of snoRD116 function. He's made great progress in year one, and these are really complex questions. This region of the genome is really difficult, and it's going to take some complex technology, I think, to really have a full understanding of what the snoRD116s are doing. And it has very similar long-term potential contributions to understanding PWS and identifying targets. The next two grants have to do with the cellular function. So how do cells, PWS cells, how do they function differently from normal cells? So not looking at their genes, but rather how the proteins are behaving. And this is a new project from Dr. Potts, who's at St. Jude's. But Dr. Potts has done a lot of work, particularly in the Schaff-Yang field. So you may remember that Schaff-Yang syndrome is a syndrome that results when the MAGEL2 gene is mutated. So in PWS, MAGEL2 is lost, it's deleted or inactivated, but in Shaft-Yang syndrome, it's mutated. And so Dr. Potts discovered that the MAGL2 gene, the protein MAGL2, is important in this normal process by which cells take receptors that are out on the surface of the cell, bring them in, kind of check, make sure they're okay, and then recycle them back out. And that process is assisted by MAGL2. So when MAGL2 is mutated in Shaft-Yang syndrome, that process gets disrupted and the receptors don't get back out onto the cell surface the way they should. And that's important because the receptors are out there to gather signals from the environment and tell the cell what to do. So they need to be out there. So he identified this in Shaft-Yang cells, and it's very likely that the same thing is happening in PWS cells, which have lost expression of MAGL2, but it might be slightly different because a mutated MAGL2 may behave differently than if MAGL2 is completely missing. And we need to understand that difference because people with Shaft-Yang syndrome aren't exactly like people with PWS. But there is significant overlap. So how can we explain that? So this project will use, and thank you to those of you who have sent baby teeth to Dr. Ryder, because this project is using the neurons derived from those baby teeth. So Dr. Ryder, who is also in Memphis, but at a different institution, but this project brings these two investigators together. Dr. Ryder has taken baby teeth from individuals with PWS and used that to grow out neurons in a dish. And so these two investigators will use the technology and approaches that Dr. Potts has used in the Shaft-Yang cells and apply them to the neurons from the PWS individuals and examine whether this, what's called an endosomal trafficking error, is present in PWS neurons. They're also going to look at subtype differences. They hypothesize that maybe the defect is more significant in individuals with PWS who have autism. And so Dr. Ryder has kind of a bank of cells, some with autism, some from PWS children that had autism, some not, and they're going to look at differences there. And they're also importantly going to see, using genetic techniques and other techniques, whether they can rescue that receptor trafficking. So can they overcome that defect? So this is a really exciting grant because it defines a cellular phenotype using their unique resources that have been developed by this team. A good cellular assay would be important if we want to do drug screening down the line. It establishes a new collaboration between two outstanding investigators with complementary expertise. And in the long term, it really gets at the molecular cellular defects in PWS neurons, and it establishes a good system by which we might be able to do drug screening in the future and identify new therapies. A second project looking at the cellular biology and the role of MAGL2 in PWS and Shaft-Yang syndrome will be performed by Dr. Rachel Weverick, who has a long history of studying this gene and this protein, both in PWS and in Shaft-Yang. And she has new evidence that one part of this protein, the N-terminus, or kind of the left-hand part of it, has a role in binding and stabilizing RNAs, and that loss of MAGL2, either by PWS or a mutation in MAGL2, disrupts this process and may change the stability of these RNAs. So she is going to characterize this MAGL2 protein complex that binds to these RNAs in neurons and in muscle cells, and look at, validate that these complexes assemble appropriately in normal cells and not so in these Shaft-Yang and PWS cells. She's also going to look at how mutation or loss of MAGL2 affects RNA metabolism. So how does it affect how stable these RNAs are? This is a novel function for MAGL2, which could explain some of the differences that we see between Shaft-Yang and PWS. So this is a really interesting new function potentially for this protein. The investigators is experienced and has a lot of experience with the cellular and mouse models of PWS. So in the long term, this again will be contributing to our understanding of the normal function of one of the genes in the PWS region, the MAGL2 gene, and it may provide some tools and resources for doing additional genetic and drug screens to develop new therapies. The mouse model study that we're supporting in this grant program this year is an extension of Dr. Giles' work. As I mentioned a little bit earlier, he's developed this way of deleting snoRNAs after the mouse is born that allows them to become obese. But you might have caught that I said that it's a portion of the mice. So it's not all of the mice that go on to develop obesity. So in the second year of funding, he's going to refine this approach so that hopefully he'll have 100% of the mice becoming obese, and that will be just a really practical thing for if you're going to do obesity studies. We want all of the mice becoming obese. And he'll also do a more detailed examination of the mouse characteristics after they lose this snoRNA and become obese, looking particularly at their hypothalamus. So this builds on encouraging findings from year one, and we're excited about it. This is at the University of Cambridge. They have a very nice group that they're developing, kind of a critical mass. You may remember that Dr. Holland is also at Cambridge. So kind of building this group of PWS researchers at Cambridge is a nice advantage of this grant as well. And, this mouse model that recapitulates the obesity phenotype in PWS is really a resource that's needed for the field, which will allow better drug development and also allow us to look at existing therapies and test them out in a new PWS mouse model. I mentioned that we're going to be looking at the gut microbiome, and this is, again, a mouse model study using the MAGEL2 PWS mouse. And this is a new investigator to PWS, Dr. Kosta Matali from Baylor University, and Matoli. And his lab has previously demonstrated that there's a critical role for the microbiome in the social behavior of mice. So he was looking in autism, mouse models of autism, and showed that if you fed mama mice a high-fat diet, they developed a microbiome that was kind of suboptimal, and that the offspring of those mice had impaired social functioning. So, and specifically, he's a neurobiologist, so he showed that it is dependent on the oxytocin pathway. However, he could restore social functioning and oxytocin functioning by replacing one of the bacterial strains that was missing in the gut microbiome. So he's really interested in mouse models or human autism disorders that are contingent upon oxytocin functioning. And of course, PWS fits this bill very well. We know that oxytocin is decreased in PWS and in the brain, and that oxytocin is a potential therapy for PWS. So he became very interested in PWS, and we became very interested in the microbiome. So this is a nice bringing together of those interests. So he's going to use the mouse model of PWS to look at how their microbiome is different than normal mice, and test whether restoring a different microbiome can impact their oxytocin functioning and also their social development. So this is an exciting grant because the gut microbiome is being increasingly recognized as an important what had been an overlooked aspect of health and well-being, and the fact that you can manipulate the gut microbiome and have an impact on health is very attractive. This group is leading in the field as far as looking at the interaction of the gut microbiome with behavioral aspects. And in the end, there's a potential for a very accessible therapy if we, manipulation of the gut microbiome to improve social behavior. The final grant that we'll be funding at this time is a project from Dr. Elizabeth Dykens with Elizabeth Roof at Vanderbilt University. And this, again, is an extension of an ongoing grant right now looking at improving social functioning in PWS, not with microbiomes, but rather by learning. So Elizabeth Elizabeth and Elizabeth have had a long-standing interest on the well-being of individuals with PWS and their families. And one thing they've noted is that as people with PWS hit adolescence and adulthood, oftentimes they become very lonely and socially isolated, and this can really contribute to the mental health problems that are already present and at high risk in PWS. So they had the idea of a web-based program that would bring people with PWS together and help them learn more social skills. Our people with PWS really, they want to be socially engaged. They want to be socially accepted. They're interested in learning how to become more socially adept. And so far, this program has enrolled more than 50 individuals in a directed curriculum to improve their social skills problem solving, to help them think about ways to function better socially within their family and with friends. And so this has been a very productive program in year 1, and in year 2, they'll be completing the intervention, evaluating the data that they have, showing whether what these individuals learn in the setting of the web-based curriculum is translated into kind of their everyday living. And their ultimate goal is to develop a curriculum that can then be disseminated and used widely for individuals with PWS in particular, but also potentially for other intellectual disabilities. We're really excited about this grant because social isolation is a huge problem in adolescents and adults with PWS. And, to give them the skills that they need to function better and be more engaged in society is a really critical aspect. The potential long-term contribution is to have this validated social intervention program that's tailored to people with PWS to help them cope and integrate more effectively into society. So that's the grants that we've approved to date. I'll mention that we have one more grant pending that we're looking to collaboratively fund with some other organizations. So you may hear about that coming up soon, I hope. What to expect in 2018? Well, our grant program continues to be quite robust. We have 25 new projects that are going to be under review that are due May 10th, and those will be announced in the fall. Our directed research programs are also moving along at a good clip, including our clinical care, genetic therapies, pharmacotherapies. You can expect this year that we'll continue to advance our basic knowledge of PWS and identify and move forward with new targets for intervention. As I mentioned, we have many interventions that are coming into clinical trials, and I really encourage you to sign up for the May 15th webinar, to check our website, to start looking at these clinical trials and thinking about whether one of them might be suitable for your family and your loved one with PWS, or the other ways that you can get involved as well. The clinical trial consortium activities are looking to support these clinical trials and help bring the voice of the PWS patient community to the clinical trials and to the regulatory agencies so that they can understand the needs in our community. So you can keep up to date with that, which is, the consortium is a consortium of patient groups, industry partners, academic investigators, all advancing clinical trials, and you can look at that website. And expanded use of the global registry to support research at all stages. So please register in the global registry. We will send you, if you're eligible for clinical trials, we'll send you notices through the registry. To that effect, you'll have opportunities through the registry to participate in, we're developing in collaboration with an industry partner, we're developing a new instrument to measure anxiety in PWS, and we'd like everybody in the registry to participate in evaluating that so we can get a sense of how well it works in a large population of individuals. So there's lots of ways, again, that you can be contributing to the overall effort of moving clinical trials forward, even if you're not currently, enrolled in a clinical trial. And we'll continue to engage and hopefully take leadership roles in the rare disease community through our partnerships with for example, NORD, the National Organization for Rare Disorders, our work with the Clinical Trials Transformation Initiative, and other rare disease and clinical trial initiatives. And finally, we'll see you hopefully in Las Vegas. Please mark your calendar for the FPWR conference in October. We hope to see you there. There'll be a lot of updates on the research. There'll be many of the industry partners that are doing clinical trials in PWS will be there to answer your questions directly about these clinical trials, so you can get more information and learn more about that. So we hope to see you there. So here are some of the websites that you can go to keep up to date on everything that's going on in PWS research. You can always email me if you have particular questions about the grant program. Jessica's a great resource as well.